Dr. Barb Rohrer on drug therapy, identifying a drug or compound to make retinal cells stronger.
I am trying to help the cell protect itself by making more energy. The big problem with not only diseases but also aging is that an aged cell makes less energy. It can't make enough energy when it's stressed. And so a disease is a stress condition. So you need more energy to combat all the problems that are generated by a defective gene. And in order to do that, you need a lot of ATP, you need a lot of energy to do that. And so if I can increase the cell's endogenous ATP production, I can allow the cell to try and repair itself to some degree. I will not be able to restore vision, but I can prevent the degeneration or the progression of the loss of vision. You start with a large number of compounds, and in our case, we had started with 50,000 compounds that are part of a library that our drug discovery core at our school has purchased. And those 50,000 compounds were pre-selected to be drug-like molecules, and so they're all small molecules, so that they're all available in the open market. So everybody can use them. And so we basically designed a cell-based method, how we can, in a high throughput manner, which high throughput means that you can use 96-well plates or even larger plates with larger number of wells that you plate cells into, and you have to have a cellular readout that you would like to test these compounds on. And we chose a cell survival assay, and as the stressor, we used something that's very important for photoreceptor, and that is calcium. And high levels of calcium are toxic. So we basically generated a cell-based assay screening these 50,000 compounds for their ability to protect against calcium cytotoxicity. And after that, we can then move the compounds that basically come out at the end of this screening tool, and then assay what kind of pathways, by which means do they protect against calcium cytotoxicity. What we asked is whether they increase mitochondrial respiration. So do these compounds protect the cells by increasing ATP production so that the cell can maintain homeostasis by itself? The reason why we chose that is because retinitis pigmentosa involves over 50 genes with hundreds of mutations. So while gene therapy might be doable for very rare cases, the majority of patients will not be able to benefit from that because we can't really develop hundreds of different types of gene therapy for these patients. So in the short run, while we're trying to protect those cells from dying, we argued that we should be able to use increased energy production for these cells so that they can protect themselves. And that was the basis of this of our first high throughput screen. The second mechanism, or the second step then, is to use the compounds that came out of this screen and then test them for their ability to increase mitochondrial ATP production, as I said, and then move it into animal models of retinitis pigmentosa, of which we've used a number of them, both in organ culture, where we use the isolated retina and ask whether a compound can protect the isolated retina. And the reason why we did that was that we don't need to rely on the ability of a compound to get from the cornea to the back of the eye, or be orally available or injectable, or whichever way we would like to deliver it. So we get it straight to the retina. And so we basically went from a cell to a mechanism to an organ culture to an animal to try and move through the different steps of increasing complexity to show that we have efficacy.